I, Shivanshi Gupta, welcome all of you to the afternoon session of today's training schedule. The first lecture we have is on quality spawn production techniques on mushroom cultivation. It is to be delivered by Dr.Mohammad Najeeb, Associate Professor, Division of Plant Pathology at Faculty of Horticulture, Shalimar, SKUAST-KASHMIR. Dr. Najeeb is associated with Mushroom Research and Training Center from his early career. He is also involved in the establishment of MRETC at Faculty of Agriculture, Wadura in 2018. He has many, that is 73, national and international publications to his credit. Also, he has authored 2 books on button mushroom cultivation, which are released by the SKUAST-KASHMIR, Shalimar. Currently, he is guiding 3 masters and 2 PhD students. Sir, I request you to present the lecture.
Professor Dr. Ali Anwar, Head Division Plant Pathology, Dr. T.A. Wani, and Dr. Tanveer sir, and dear students, Assalamu alaikum and a very good afternoon to you all. At the outset, I would like to thank the organizer of this training program, especially Dr. T.A. Wani, for giving me the opportunity to interact with you all and for giving me the opportunity to share my little experience with you. Today I have been asked to deliver a lecture on the quality mushroom spawn production technology. As we see, mushroom is an emerging agriculture enterprise, not only in the world but in India and in Jammu Kashmir also. Many young fellows, whether educated, uneducated, they are getting involved in this activity because of the high returns, obviously. For this mushroom to be successful, there are certain requirements, and first and foremost is the quality spawn. The spawn is actually the seed of the mushrooms. Like we have in agricultural crops, wheat and rice, we have seeds which are produced by the plants and which we use to propagate the crop. In case of button mushrooms, we have spawn. In case of all the mushrooms, we have spawn. Under natural conditions, the mushrooms multiply by means of spores, which are produced in their gills, and they are taken out by the air currents and spread to distant areas, and then the mushroom continue to grow naturally. But when we try to cultivate mushroom inside at our own disposal, at our own time, we need spawn. This spawn is actually the substrate of mushroom fungus, which is covered by mycelium of the fungus. So, before deliberating on the lecture, I will give outline of my lecture. First, the introduction, infrastructure required for spawn production, mother culture preparation, preparation of spawn, commercial spawn production, and methods of spawning and precautions in the spawn production which we need to take care of so that we can produce quality spawn. As I told you, like in agricultural crops or horticultural crops, we need the propagule to produce the crop. Similarly, in button mushroom, we need spawn to multiply, to propagate, to cultivate the mushrooms. This propagating unit consists of the substrate plus the fungus of the mushroom, which is fully colonized by the mushroom fungus. And then when it is fully colonized, we call it spawn. A little bit of the history. Before, as it is seen naturally, in the history, there was no spawn production. People used to gather the mushrooms from the forests, from the pastures. But as the time evolved, mushroom when become the business, we need to produce the spawn. Up to, it was first Culpeper in 1692 who described spawn as mycelium of the mushroom. And up to 1894, spawn was gathered from the wild. It was not as a business. It was gathered and then sold, but it was not produced by humans as a business entity. And before this grain spawn, which is today universally known as the best spawn of the mushroom. What I already told you, the wheat grain spawn of the wheat grain, that is considered one of the best spawns. Before it was started, there were other spawns that were used, and among them was flake spawn, which consists of the bread on which the mycelium of the fungus was grown, and then these flake were used as inoculants for the compost. Then there was another spawn, mill spawn, where they prepared the bricks of the horse dung or cow dung and the loam soil that was inoculated by the mycelium in the nature, and then that was used as spawns. Later, as the time evolved, then manure spawn was used. This was first spawn that was sterilized horse dung or compost that was inoculated with the mycelium of the fungus. The first pure culture spawn was produced by Constantine in France in 1894, and Duger in 1905 prepared pure culture mushroom spawn, which was later used to inoculate the bottles. The first grain spawn was produced by Pennsylvania State University in 1932. So, before venturing into the spawn business, before venturing in the preparation of the spawn, it is important to know what are the infrastructure and equipment requirements for spawn production. This is the picture of layout of the building, which we call as spawn unit, spawn producing unit. It is not necessary that when you start the business you have to consider all these structures. Initially you can start in single room, which will be which should have the laminar air flow, the autoclave, and the storage room. But when you are doing it commercially, if you want to establish the spawn room, spawn production unit, then you have to have these components of the spawn producing unit. Here you can see this is the entry, then there is the lab. When you enter into this lab, before that you have to change your clothes in this changing room. Then you enter the lab. In the lab you have store where you have various requirements for the spawn production. Then there is boiler room, and then in the lab you will dry these grains on the table, lab table. Then you have autoclave room where you will autoclave and sterilize the spawn. And after autoclaving you will find here the inoculation room. This one is the inoculation room, where you inoculate the sterilized bottles with the mycelium of the fungus, and after inoculation you incubate these in the incubation rooms. This is just an outline, but you can always modify it as per your availability of the place. Then there are minimum number of equipments that can be used. It does not involve very high cost, but still up to 5 lakhs, within 5 lakhs you can establish a spawn producing unit. This one is the utensil used to boil the grains. This is the autoclave, horizontal autoclave. This is the laminar air flow, and this one is the BOD incubator. I think all of you are aware, we have just been in the practical and you have seen all these equipments. Then I told you that in general fungus, the propagative material comprises of the mycelium as well as spores, and in case of similar holds true in case of the button mushroom. In case of general fungi, we have asexual spores and sexual spores, but here in case of the button mushroom, we generally reproduce it by the vegetative means, by the mycelium bits, or we can have the sexual spore, that is the basidiospore. But commercial mushroom is always prepared from the mycelium bits or tissue bit culture method. Then for raising the pure culture, we require tissue culture. We get the tissue from the collar region of the button mushroom. Collar region is the region where the stalk and the cap combines. There is a thin boundary which we call collar region. We take the tissue from there, and then we put in the sterilized media, incubate it, and we get the master culture. And when the master culture is prepared, we use that master culture to prepare master spawn. There are two terms, master culture and master spawn, which I already made clear in the while practical, but you still master culture is the culture prepared directly from the fruit body of mushroom, whereas master spawn is the spawn produced from the master culture. The spawn which we produce from the master culture, that becomes the commercial spawn. If we need to purify the culture, we can use it by subculturing, which is done regularly. And to grow the this mycelium, this fungal culture, we need the different medias. We can use different medias. These are the list of medias: potato dextrose, agar, oatmeal, agar, compost, agar, wheat extract, agar, and rice bran decoction medium. We can use any of these mediums and prepare our master spawn. But in general, potato dextrose agar is preferred for cultivation of this, for preparation of master spawn. Then this is the demonstration of the tissue bit method for preparation of master spawn. As you can see, this is this picture is the inverted upside down button mushroom. Here you can see the second on the right top. Pointer hai or
Pointer nahi laya sir.
Achha. So you can see the second picture where the top of it you can see that from the collar region the tissue bits are taken and then they are inoculated in the petri plate, and then you see the master spawn, master culture in the petri plate after incubation. Then this is the spore print method where you get the spores from the fruit body. Although this fruit body is not agaricus, this is some other mushroom, but the spore print is obtained when you place the fruit body cap of the fruit body on the plain paper overnight, and in the morning you can see the print like this. The disc shape, this one and this one, they are the support prints. And then you can use these supports for inoculation and for getting the master spawn, master culture. But there are some mushrooms which are heterothallic, which will not grow from a single spore. They will need another spore, and then they will combine, and then they will produce the fertile mycelium. But in case of button mushroom, we are lucky enough that even a single spore can produce the pure culture. So these are the methods how to streak them, how to make the pure culture. Then the maintenance of the culture. Once you get the master culture, you can use it for preparation of master spawn. But maybe there is a time lag between, so you can preserve this culture ordinarily by keeping it in the refrigerator at 4 degrees centigrade and use it when you are free and when it is needed. There are other methods of preserving the culture which I will not go into the details. This is long-term preservation of the culture. You might have gone through it in the last lecture yesterday. So I will not be deliberating on these methods. Then the spawn production, which is the main activity here. After this needs a substrate. As I told you, there are many substrates which can be used for preparation of this spawn. The most preferred one is the wheat grains. And next phase is the preparation of these substrate for the inoculation. We use the healthy seed grains. We boil them for 10 to 15 minutes, drain the excess water, and then dry these, and then calcium carbonate and gypsum is added as we have shown you in the practicals. Then after putting in the bottles and bags, we autoclave it for 121.4 degrees centigrade for 2 hours. And after autoclaving, we inoculate it with the master spawn, and we get the master culture, and we get the master spawn after 10 to 12 days. Then, as I told you, this master spawn is prepared from the master culture directly. And when this gets ready after inoculation, this takes 10 to 12 days for completion. And once we get the master spawn, we can use it for the further multiplication for the commercial spawn. This is the general outline how it goes. First the pure culture, then the master culture, then the master spawn, and then commercial spawn. This is in the figure you can see on the right side of this. And this is on the left, on the right side is this. On the left side, you can find all the procedure and the diagrammatic representation of this whole process. Then how do we know that the spawn which we produced is a quality spawn? It should have some characteristic, it should have some qualities. First of all, when you are purchasing or when you are getting spawn anywhere, you should ensure that it is of single color. We have white button spawn which is white in color. In case of Pluritus DJ mode, it is pink colored. But here the point to consider is that the spawn should have one color. If it has more than one color, that means there is some contamination. There is some other things in addition to the spawn which are not preferred, which are not liked. So it should be of one color, it should be pure. It should be fresh, as when the old spawn is used, it will result in less yield. So preferably all the times you should have the master spawn and from it prepare spawn further and use it for the production purpose. Then whatever spawn we are preparing, we should ensure that whatever spawn we are making, it should be of high yielding strain. It should give us high returns. And it should be free from contamination and it should be available at the cheap rates if you are purchasing it from the market. Then spawning is a process of mixing the spawn with the compost. The process is known as spawning. There are various methods: top spawning, spot spawning, thorough spawning, and multi-layer spawning. Top spawning we do when our compost is having high moisture. We place the spawn on the top of the bags which contain the compost, and the rate of use of spawn is 0.5 to 2%. And in the spot spawning also, we place the spawn at certain spots and then incubate it so that it can colonize the compost. Thorough spawning is another method where we mix the spawn with the bulk of the compost, general mixing. Like we mix calcium carbonate and other gypsum with the grains. Similarly, with compost, we are mixing the spawn. That is called as thorough spawning. But most preferred type is multi-layer spawning, which is practiced in Kashmir and most of the places in India. We place layer of compost in a basket, in a bag, or in a tray, and then place one layer of spawn. Then cover it again with a compost layer, and then place another layer of spawn. The purpose is that the whole compost should be colonized by the fungus, so that the spawn run is proper. When the spawn run is proper, the yield is expected to be higher. If the whole compost is colonized, naturally we will get better yield. And then there are the precautions. Some of the precautions while preparation of the spawn is, as in most of the crops, in all agricultural crops without any exception, we have the problem of diseases. Similar is the case with the mushrooms. If we don't take care, we don't use cleanliness, we don't use sterile material, we will be having the contamination, we will be having the diseases. So cleanliness is most important for maintaining the disease-free spawn. Then we have to follow the proper procedure for during the spawn production, and we have to sterilize our hand by using various materials, by citrate, etc. And we have to sterilize the equipments. Before using the laminar airflow, we have to switch on the light for 20 minutes, and then after 20 minutes we have to switch off the UV lights and switch on the tubes and air, so that the outside material, outside air, outside contamination does not enter into the laminar flow. Similarly, we have to use disease-free and healthy seed grains. We have to see that the grains are not overboiled and grains are not underboiled, both of which can create problems. And we have to use always sterilize the bottle properly or bags properly for proper duration of time. And while we are preparing this spawn, we have to shake the bottle 3 to 4 times so that the time taken in the spawn run is less and we get the spawn that has fully colonized the grains. And after the spawn is ready, we have to store it at 4 degrees centigrade. That can be in the refrigerator, in the lower shelf of the refrigerator, which has 4 degrees, we can store it. Then one of the important thing is that we should use always fresh spawn. if that is not possible, we should use maximum 3 to 4 generations, not more than that. That will affect the yield of the mushrooms. Then when we are using the spawn, we have to label the bottles with proper date, time, species and other things so that we can know the age of the spawn. And this is all about the spawn production. I was told to finish this in a short time, that's why I was...
Okay, thank you, Dr. Najeeb. Nicely you presented your lecture.